of the basic techniques of plant biotechnology, that is plant tissue culture. What is plant tissue culture and how it is done as the technique. And we saw that how following those protocols, it is possible to now culture any part of the plant in vitro, and successfully you can culture these intact plants, or you can go for embryo culture, organ culture, shoot tip culture, callus culture, cell suspension cultures, protoplast cultures, So it is now possible to not only keep these cells, tissues, organs alive in the in vitro systems, but also you can regenerate whole plantlet from the somatic cells and tissues of a plant. And then I told you that there are many applications which have been, these techniques, basic tissue culture techniques, have been exploited for various applications in terms of using these techniques for manipulating or improving the plant quality. And for example, you can see micropropagation is one application of basic tissue culture technique of somatic embryogenesis and organogenesis, germplasm preservation, somaclonal variations, dihaploid formation, and then ultimately genetic engineering. Now, genetic engineering also uses this basic ability of plant cell, somatic cell, to get regenerated into whole plant. Now, there what is done, you take the cells, group of cells or single cell, and genetically modify and then regenerate plant from the modified cell, and you end up with genetically engineered plant or genetically modified plant. In this session, very specifically, I'm going to focus on the application of tissue culture techniques for micropropagation or clonal propagation, interchangeably used, both means the same, and germplasm preservation, how the basic tissue culture technique has been exploited for these two applications. So first, what is clonal propagation? So basically, if you are familiar with the mode of reproductions in biological systems or in organisms, you have asexual mode of reproduction and the sexual mode of reproduction. So sexual mode of reproduction takes place through involving the sex cells and forming zygote, and the zygote undergoes division. And asexual mode of reproduction involves the somatic parts of the plant, of the organism. And generally, asexual mode of reproduction is seen in plants invariably, and prokaryotes, and lower animals. But higher eukaryotes, higher plants, they undergo both, plants undergo both asexual and sexual reproduction, and animals, they undergo only sexual reproduction. Okay. So clonal propagation or micropropagation is a kind of asexual reproduction, where where a new plant is generated from somatic cells. And advantage here is that the new plant has the same genotype, because the genetic variability comes through sexual mode of reproduction, and So one of the advantage of clonal propagation is that you get genetically identical plants following asexual mode of reproduction. You know. So as I told you, what happens in sexual reproduction, you have the sex cells fusing, forming a zygote. Zygote undergoes multiple divisions and then differentiation, maturation, and then you get the new plant-like formation. So that is the normal zygotic mode of reproduction. But then in micropropagation involving asexual mode of reproduction, you have three possibilities by which you can get the new plantlet formation. One is through embryogenesis. Now, this embryogenesis is not the zygotic embryo, which I explained to you in previous slide, but this embryo is differentiated from somatic cells, that is by direct embryogenesis or indirect embryogenesis, means involving callus in between or directly from the explants. You have the embryonic formations, and you can see the embryo-like structure being formed, you know, and these embryo-like structures they resemble with the zygotic embryos, you know, so in the structure and also of course function because they can undergo the germination process to give rise to the new plant. So one is embryogenesis mode of formation of new plants, and another is organogenesis. Now this organogenesis is, as I explained to you in my previous session also, it can be via callus, means you have the explant, it undergoes de-differentiation and gives you a mass of cell, which we call callus. And then this callus undergoes shoot formation and root formation, what we call the organogenesis. So that is one way of organogenesis. Another is without involving callus formation, and you have direct organogenesis from the explant. So you take the explant, put it on a media, give appropriate conditions of growth regulators, and you can have the direct shooting and rooting. And the third one is the microcutting. Microcutting is basically the normal process of cutting way of propagation of tree species in the ornamental plants. This is routinely done through cutting process. Here what is done that we take the meristematic zone, the actively dividing zone, and like the shoot tip culture or the bud cultures, from the whole cutting, we take the very small part and then culture it, and that is made to give rise to the intact plant, using tissue culture techniques. So basically, both somatic embryogenesis and organogenesis, both these processes happening in vitro have been exploited for clonal propagation purposes or micropropagation purposes. And now what happens? Both these techniques involve a longer exposure of tissues to growth regulators. And therefore, most of the time, you don't end up with having genetically identical populations if we go by these methods. Okay? So that um, but then most effective way of producing multiple plants using this technique is starting from a single cell. So or a few cells. So if you start with single cell or from few cells and then make them to undergo somatic embryogenesis or organogenesis, then probably you can end up with better plantlets as compared to I mean, that's the way you end up with multiple plantlet formation. So the point I raised, that the longer exposure of the plant cells or Kali cultures uh, does not always give you genetically identical. So if the effort is, if we can reduce this period of exposure of these explants or the careless cultures to growth regulators, that helps in getting the genetically identical plantlets in this, because that is one of the objective of micropropagation, is to get genetically identical offsprings or the plantlets from the source. So we don't want that to change. The genetic makeup of the original plant should not change. Okay, so so what is then somatic? We will go a little bit in details into the somatic embryogenesis. So it is the formation of embryo-like structure, which we call somatic embryo, from single or group of somatic cells. Okay, so therefore it differs from the natural sexual mode of reproduction. Okay, so this is what is done in somatic embryogenesis, and mm, and somatic means, since it is derived from somatic cells, so somatic embryogenesis, embryos are genetic, and they are analogous to zygotic embryo basically, because they resemble in their function and structure also, and all the plantlets derived from here, from this, are genetically identical. They have the same genetic makeup. Pictorially, if you see here, it is the single cell or the group of cells. They on the media, they are allowed to grow or multiply, and then they undergo the cytodifferentiation of globular stage. These are different stages of embryo, which is seen in natural zygotic embryos also, the globular stage, heart-shaped stage, and torpedo, and then you have the plantlet formation. This you can see, which happens in zygotic embryo, this kind of development the embryo undergoes during sexual reproduction, that zygotic embryo development is seen in the somatic embryo development also. So that's how they look like. This is the globular stage, heart-shaped stage, and torpedo stage, and these are the germinating cotyledonary stage. Uh, so this is how a somatic embryo undergoes the development, and this is seen in the alfalfa plant or Medicago sativa, somatic embryos developing in the developmental stages. Uh, or in peanuts, for example. This is how the somatic embryos they look like, and they are put into the suspension cultures, and finally you see the plantlet coming out from the somatic embryos. So this is the further growth of these plants into thing. So what are the benefits as I've been pleading for as an alternative tool for multiplying tree species of the ornamental plants using micropropagation or the clonal propagation pathway. So that is the variability which arises out of sexual reproduction is not there in micropropagation, okay, because it is done through somatic cells. And genetic variability which is seen in nature is because of sexual reproduction. Secondly, the seeds generally show dormancy, means after the seed is produced, it undergoes a period of rest, what we call dormancy. Now, that is also a negative aspect of in propagation. If you have a seed which is taking 6 months, 1 year as a dormancy period, as against you have these somatic embryos pathway which can be immediately, germinated and can give you a plantlet um, immediately. And also you can get plants free from the pathogens or the infections, because the source, if the source is purified or pure, if you have made sure that the source plant is healthy and you are taking the somatic tissues from there, then you can have a disease-free plant. So these are some of the benefits of micropropagation or clonal propagation as against the seed multiplication. You know. Now, the second aspect which I want to cover here is the germplasm preservation. Now, what is this germplasm preservation I'm referring to? We have certain endangered plants, means those who are at the verge of extinction. Now, how we can preserve those plant species using this method, what we call artificial seed formation, and also artificial seed or the germ plasm preservation has advantages. First, let us look at what are the limitations when it comes to uh, propagation through or preservation through normal seeds. You can just collect the seed and preserve, and that's how the germplasms are preserved. So, but then that has some limitations. That is how to maintain the genetic variation in a normal seed stock preservation if we are preserving seeds directly. So how the genetic variability maintenance is a difficult factor. Fruit crops are difficult to produce seeds. If you see the fruit trees, they take longer time, and the seed production it takes time. Okay, and seed-borne diseases are there. So if you are preserving a seed which is carrying the disease material with it, then it is not going to be helpful. And then the dormancy factor is there, that seeds have normal dormancies before they again undergo germination and give rise and make a plant. You know. So what? So the synthetic seeds, what we call a new term which has come. So what is the synthetic seed means, and how it is better than the preservation via normal seeds. So basically, synthetic seeds is living seed-like structure derived from somatic embryos in vitro culture after encapsulation by a hydrogel. So what is artificial seed, synthetic seed or the artificial seed, where you take the somatic embryos and encapsulate it with gel-like substance. Okay. Uh, And this gel like when you cover the somatic embryo with gel-like structure, it protects the embryo from microbial attacks, things like that. Okay. So normally what is done here, the somatic embryos are artificially encapsulated, and and shoot bud, cell aggregates, or any other tissue, can be used for generating somatic embryos, and then uh, these somatic embryos are able to get converted into whole plant. Once they are put for germination, you know. So basically, they retain their ability to undergo germination even after storage. The storage can be for years you can store. So that's how a synthetic seed looks like. This is the somatic embryo, and this is the gel in which it is embedded. You know? So this is how it looks like. So So what is the method of generating this? So first you have to establish the callus culture and then induce the somatic embryogenesis in that, then mature the somatic embryos undergo maturation and then encapsulation of these, and uh, these seeds are tested, and then they are they're tested for their ability to get converted into plant, and where they're taken from lab to greenhouses and then to the field. So this is the total outline, of the activities of which are involved in producing synthetic seeds. Okay, that is starting from establishment of culture, then and induction of somatic embryos, maturation of embryo, encapsulation, and the test of synthetic seeds. That is by germinating them in lab, taking the plant to greenhouse, and then to the field. When your stock, artificial or synthetic seed stock, undergoes all this, it means you are ready for preserving these synthetic seeds for future use. You know? So this following this, a lot of success stories are there for different crop plants, where the in vitro propagules have been encapsulated, like somatic embryos for papaya, brinjal, mango, carrot, sandalwood, brassica, lettuce, auxiliary buds, etc. in these plants, eucalyptus, citrus, pineapple, shoot tips encapsulated in case of banana, cardamom, apple. So there are many examples where the synthetic seeds have been generated and used for store. Oh, so now you understand that somatic embryos are generated. Now how they are encapsulated. Next question. And these are different gelling materials which have been tested successfully. Sodium alginate, sodium alginate with gelatin, all this. And these are the complex ingredients, and this is the final concentration. So all these agents have been successfully used for encapsulating the embryogenic tissue. So this is how the somatic embryos are encapsulated. That is by this is the droplet method. So you have the gelling material here, passing through this funnel, and you insert the embryo, somatic embryos here, and then it is complexed here, and then these are washed. So simple method of coating the gelling material around the somatic embryos, and they look like something like this. So pearl, like pearl. So this is in banana actually. This green structure, green dots, what you see is the somatic embryos, and the white is the gel material. So again, this is part of micropropagation, generating synthetic genes for storage purposes. Here you can take each one of them, for example, like this. You can put on the growth medium for germination, and then you can see that they are germinating, shooting, rooting, and they are transferred to the pots, and then taken to the field. So, and these plants, the added advantage is that they have the genetic makeup of the mother plant from where the somatic embryos have been generated. So taking, we are able to propagate same genotype, using this technique. Okay, so Synthetic seed is an extension of micropropagation, wherein you can store these embryos can be stored for a long period of time. So, to conclude, what is the advantage of this? So, so we can directly take the seeds or tissue culture derived seeds material to the field. It's like any other seed material. You can directly put in the soil also, and there it will germinate. And then we can have the desirable genome organization or the genotype or the genetic uniformity from the mother plant from where they have been derived. And then the cost is reduced, you know, by vegetative propagation, you know, and the germplasm can be preserved like this. Dormancy is reduced or it is not there because it is from the adult. So you take the antisomatic tissue, so it can directly germinate. There is no dormancy period involved, as one has seen in case of artificial. I mean, the natural seeds. All natural seeds are dormancy. So that problem is not there, and you can produce large number of these kind of seeds um, in a very short period of time. You know? And it can be produced throughout the year because it is done in the lab. So you don't require a particular season where the plant can grow and you can then propagate it. So you can keep on producing these seeds all through the year. So with this, uh, mm, you have seen that how basic tissue culture technique of somatic embryogenesis particularly has contributed immensely for clonally propagating or asexually propagating the tree species, particularly, and the ornamental plants, and also has contributed in preserving the germplasm via synthetic seeds. So